Hi, everyone, and welcome to this session of the Situ Q&A with Dr. Jonathan Kolby. My name is Sophia Wood. I am Opwal's country manager for the Ecuador programs, and I also run Friends of Wallacea, which is an initiative we started to help Indigenous communities that work with Opwal to develop wildlife tourism that promotes conservation. Today, we are lucky enough to be chatting with Dr. Jonathan Kolby, who is the Fish and Wildlife Society's policy specialist and a frog conservationist who's worked on several projects on the Honduras project with Opwal and Casuco National Park for nearly 10 years. And we'll have a chance to chat a little bit with him about his experience researching chytrid fungus in amphibians around the world. And we have lots of questions that have come in. So if you have any additional questions that come up during our chat, you can send questions via email to xc2@opwal.com, even throughout the talk, or you can send them via the chat on YouTube. If you're watching this through the Opwal website and you can't see the chat, click on the YouTube button and it will open up in a separate tab and you should be able to send your questions. So thank you so much, John, for joining us this morning. I've got lots of questions for you, so
Thanks. And yeah, thanks to everyone for tuning in and being interested in the work that I've been doing. I will just mention one really quick thing. Thanks for that wonderful introduction. But I do need to mention that I am a former Fish and Wildlife Society's policy specialist. I did recently leave that position to be freelance and do more of this conservation work. But yeah, I'm legally obligated to say former Fish and Wildlife Society's policy specialist.
fantastic. Well, sorry for that.
Well,
So we got lots of questions about your work studying chytrid. So there's, I think right now, studying obviously pathogens and how they spread between humans and animals is something that's really prescient and germane in a lot of people's minds. So I wanted to ask you kind of, how do we prevent zoonotic diseases from infecting us, especially when we're doing wildlife research in undiscovered areas where we maybe don't know what exists?
Yeah, really good, timely question. There's a couple different answers to that. I think overall, as we're learning from the whole COVID-19 situation, that we truly need to recognize the close connections between animal health, human health, and environmental health, which we call One Health, which is beginning to gain more attention and recognition. And when you start thinking in that kind of mindset, it's easier to identify risks that you might be putting yourself or other people or wildlife in when coming into close contact with them, whether it be, through handling, as a scientist, we always have to take precautions, wearing gloves, sanitizing our equipment, and doing what precautions we can, to more casual interactions that we might not have otherwise thought of as risky. I think it's a good point to raise that someone asked, how do we protect ourselves from diseases that we don't know are out there? And it's kind of a catch-22. I mean, obviously we can't stop everything if we don't know how, but that doesn't mean we can't protect ourselves better. And a lot of these diseases spread in very similar ways by close contact with wildlife, and taking basic precautions can protect you from things that you don't know are out there.
Right. That makes a lot of sense. I mean, and kind of on that note, you know, it seems like obviously we're dealing with kind of a human epidemiological response right now. And one of the questions that came up is kind of, why do you think that so many countries around the world are lacking governmental agencies that screen imported wildlife for pathogens? I know this came up in your National Geographic article. But seems like that's a pretty big oversight.
Yeah, and that's something I'm really interested in diving into deeper. And I think it's to me, in my opinion, it's that traditionally, first off, we and governments and people in general don't often look at wildlife health as a high priority in our daily lives. We live largely in a bubble, separated from nature. And if animals are dying and suffering from diseases, we don't often feel like that's an emergency that affects us, that we need to invest time and money and effort into addressing until people start getting sick.
Right.
And as we've seen here with COVID-19, that's what happened again. These are pathogens that are naturally in the wild, that live and coexist with wildlife. But it's not until people start messing with that then the issue of wildlife disease becomes, an integral point of our attention. So I think a lot of countries are still in a very reactionary mode, as are we in the United States, where traditionally our culture of dealing with these things is responding as well and as quickly as we can when disaster strikes. But by far and large, I don't feel we're very proactive. We're not doing as much as we can to be doing surveillance, be doing testing, be doing monitoring for a wider diversity of species before things get ugly. So personally, I would rather not wait until we think a disease is zoonotic before we pay more attention to what's happening in nature.
Yeah.
But often that's what it takes for people to notice.
Yeah, that's tricky because it doesn't seem like chytrid is probably going to affect us directly, other than obviously this, major mass extinction of frogs, which is obviously also a huge deal. So I kind of want to hop into a bit of your story, because when you hear a story like yours in which you have this clear undying passion for amphibians, it can sometimes feel like you must have always felt that way and it was such a clear path all along. But most of the time, these things are kind of things that develop over time. So I'd love to hear kind of your process of, realizing your passion for amphibians, and how you began to develop that.
Right. So, I was one of those children that were just always running around the woods, catching, frogs and snakes and loving all that kind of stuff. So I always had an interest. But, it wasn't until I was a teenager that I really found a passion in learning more about these animals and doing field work and volunteering my time with conservation groups, with reptiles, amphibians, like all that kind of stuff. Back then, when I was 15, 16, 17, I was doing a lot of work in Southeast Asia, and specifically around Hong Kong and China, helping with some biodiversity surveys with the conservation agency. And I would say, quite honestly, I was more of a reptile guy back then. And I know a lot of frog people are like, oh no, you weren't always a frog guy. And I mean, quite honestly, no. I mean,
Right.
there's really nothing more exciting than finding a snake, even still. But
Right.
there was a turning point around 2006, 2007, when I started working with Opwal in Honduras. And it was right when I was about to go back, my second season in 2007, when I had an opportunity to take a disease monitoring class with the U.S. Fish and Wildlife Service. And I learned about chytrid fungus. I'd heard about it, but I didn't really understand what it was before then.
Mhm.
And I learned how easy it was to sample for it non-invasively, and was reading some IUCN medalist assessments about the frogs that live in Cusco and how some were disappearing and no one knew why. And it all just kind of clicked that, this would make a great survey. And the results of that season were just really scary to me because I found way more chytrid than I even thought I would find anything at all. And it was kind of that point when I had to make a decision on what am I going to devote myself to? Where am I going to dedicate my time and effort in the work that I do? And as much as I find it exciting to go from place to place and look at different animals all the time in different countries, looking around me and what other people are doing, I decided I was going to be a frog person, and that there really, at that time, weren't enough people, in my opinion, fighting for the frogs and trying to bring a voice to this issue. And so I kind of joke about it, but in all seriousness, the frogs picked me, I say, and I accepted that. So yeah, that was 13 years ago, and still doing it.
Fair enough. I mean, I think a lot of, when we start to hear these stories of people getting really, really passionate about things to the point that they're doing a PhD, there is often that moment of click, that class that you took or the paper that you read that then kind of just launches the rest of your career. So in terms of all of that, obviously, even no matter how passionate you were about studying frogs, taking on a PhD is a big deal. It's it's a long time. It's a big undertaking. So how did you know that a PhD would be the right path for you, and how might you advise someone who's thinking about a PhD in biology or wildlife research?
So I know there's a lot of mixed opinions, and I have a fairly strong opinion that I will share, but I will just preface that with, there's no right answer. Everyone is different. What's right for you is not right for someone else.
Sure.
For myself personally, for quite a long time after graduating from uni, did not feel that continuing education was really necessary for me. I was happy where I was with my university degree. I was working for the U.S. Fish and Wildlife Service. I enjoyed my job, and I was still able to do all my work in Kasuko with Upwall, during my summers on time off. I was getting small grants to support my work, and that was really important to me. Um, but a lot of friends and peers and other people I looked up to would always tell me that I should go back to school, that I'm smart enough for a PhD and things of that nature. And I didn't agree. I didn't feel like being smart enough and like just doing it because that's what people do was necessarily the right thing to do, and not
Sure.
in my case. But I always said to myself, if there comes a time and place when I can't continue to do what makes me happy, which is my research and my job, that would be a reason for me to go back to school. And about 4 or 5 years later, that happened. And basically, the turning point for me was that I started getting denied from grants. Um, I was getting a lot of small grants for my work in Honduras. And then my work started getting more complicated and more expansive and bigger projects. And I started asking for much larger grants, and I started getting denied. Um, and I will mention that I was told by certain people, part of those decisions, that it was because I didn't have a PhD. Um, so that did upset me a lot, because I would have been granted the money based on merit, but when they saw I didn't have the credentials,
Sure.
I felt like, even though I didn't feel like I needed the letters after my name, I don't like boasting and bragging that, PhD, what is that? I don't feel like I should be treated any differently because I have it now. But to the people that make decisions about funding, it made a difference to them. And that was enough reason for me to devote, 5 or 6 years of my life to be able to continue this work. Together, it wasn't just that, it was also that I had a research project on the back burner that I knew realistically was so complex, and trying to understand the global spread of chytrid and like how to make sense out of it, that the best way to really be able to focus the time and effort I needed to answer those questions would be a PhD. So both of those pieces came together at the same time. But the one biggest piece of advice that I feel I want to share with people is that just think about where you want to be and what it is that makes you happy. And if you feel like you can get there without spending another 6 7 8 years of your life in school, then continue on your path. But if you think that there are things that will hold you back, like for me, then it's definitely worth investing the time and effort. But just think about it, because a lot of people I know just go get their master's, go get their PhD because they don't know what they want to do next. And
Right.
sometimes it works out for them, but sometimes a lot of my friends end up just as lost and confused as I was, 5 or 6 years ago. And I just don't want people falling in that spot unless it's right for them.
Right. No, that's really good advice. I think obviously, some people come out of university being super passionate and knowing what they want to do, but I think a lot of us come out and need to kind of, flop around in the real world for a couple of years or 4 5 6 years and figure things out until you know what you're really, really passionate about and what you want to dig deeper on. And it seems like obviously you have this kind of clear lifelong project
Yeah. And,
working with
and, and I will
frogs.
also mention, though, that, I know plenty of people who got their PhDs or their masters in a topic that they're no longer working in, or they're no longer working in academia. And I think that's a stigma we also need to get rid of, because, despite what I just said, even if you just continue school just for the sake of it, you're still learning really important life skills that will be applicable to like anything you do. So just how do you work with teams? How do you resolve conflict? I think there's a lot more personal social skills that we learn that we don't really think about very often. And that's important too.
That's a really good point as well. I mean, obviously, a PhD is very specific, but it could be, right for a different group of people than for what you're saying. That's great. I think that kind of advice is really helpful for a lot of kind of younger people who might be watching this and thinking about a path into biology and what that looks like. So I appreciate your insights on that. I'd love to dive into kind of some of the specifics of chytrid. I know obviously you're a big expert on this. You've studied it for a long time. So what is it about chytrid that makes it so hardy? How does it live in all these different places? I mean, even being in rainwater, one would think it would get, filtered out somehow. So how does it survive? How does it get into rainwater and stuff like that?
Okay. So first, really quickly, the rainwater, because I don't often get to talk about that and what it actually means or what it doesn't. So yes, I did find evidence of chytrid presence in rainwater in a storm. And the one thing I want to clarify for the public is that it doesn't mean like all hope is lost, chytrid spreading in the rain, we can't stop the rain, so we can't stop chytrid. No. We cannot prove at this time whether or not it was alive. We just know that its DNA was there, which to me suggests it could be alive if it happens often. And it was just kind of a thought experiment to say, we're trying to map the spread of chytrid around the world. And sometimes it shows up in really weird places at really weird times, and then we don't find it again. And, the two most common go-tos that people think is either, well, amphibians just move it by locomotion, which they do, or humans brought it there, either through contaminated equipment or through transporting animals or the pet trade. And yes, that happens often too. But I just wanted to show that there could be many more different ways that chytrid is spreading that we should also think about. Because, when we're trying to explain how do we respond to it, we can only respond as well as we understand how it's happening. So to me, it's a fascinating example of just how open-minded we need to be when we think about how diseases spread in the environment. But the way I think that's happening is through waterfalls. Most rainforests you go to, and especially Casuco, there's waterfalls everywhere. And I know through filtering the river water that the rivers are just loaded with chytrid fungus because it hangs out in the tadpoles, and the tadpoles are constantly shedding live infectious spores into the water. And you don't need a very high waterfall to create a spray zone. And whenever that water gets aerosolized, who's to say there's not a horizontal crosswind that's going to carry it away? How far away that moves while chytrid's still alive is based on so many variables. But if you think about how many tropical storms you have that run through these areas, you've got hurricanes going 100 kilometers an hour. So in 1 hour, could it move chytrid 100 kilometers? Why not? It moves much bigger things than chytrid that survive. So I thought that was just an interesting, poke at the nest of how do you make sense out of this on a global scale. But as far as being hardy, I think that to me is one of the most fascinating mysteries, is that in a laboratory, chytrid often seems difficult to keep alive. So when you're growing chytrid and you're culturing it for experiments, I don't do that part myself. So I'm speaking from other people's experience, but I've read that it's often very time consuming to keep it alive and happy and prevent other bacteria and other fungi from overwhelming it on a petri dish. Yet in the wild, when you have so much more other microflora that it's competing with, it's just feisty and so tolerant. And yeah, I just find that super interesting. That the infection loads that laboratory experiments often use to infect frogs is so much higher than anything that we normally find in the environment. Yet in the environment, it's just, you know, very tenacious. So yeah, I've never really seen an explanation for that.
That's interesting. Well, there's someone's PhD project right there.
Yes, please.
So obviously you've done a lot of work kind of developing methodology for helping frogs recover and deal with chytrid through HARC and through some of your research. So first of all, kind of, does chytrid, if not intervened, does chytrid always kill the frog, or do they have a good chance of survival? And then beyond that, kind of, what kind of methodologies have you developed to help frogs have a better chance of survival and hopefully build their immune system so they can go back and breed frogs that are resistant to chytrid?
So to answer your first question, every frog is different. And that's what makes it complicated as well, both in communicating to the public what's happening and also for conservation on how do we best respond to it. So the impact of chytrid varies incredibly from species to species, and sometimes even between individuals within a species. So we're still wrestling with that mechanism of what is it that allows some frogs to survive and others don't, and whether or not that's something that we can manipulate with them an advantage. But at present, it's still, after 10, 20 years of research, we still don't have a lot of silver bullet methods genetically to try to increase resistance. But as far as overall conservation methods, so what HARC is planning to do, and why we think it would be effective, is by studying how the frogs in Cusco respond to chytrid year after year after year, and looking at multiple life stages all at the same time, every time, the trends that we find across life stages is what informs what we think will be an effective strategy. So as we conventionally understand, chytrid matches up with what's happening with tadpoles and baby frogs. So it doesn't seem to kill tadpoles. It lives in their mouth. That's the only place they have keratin. So they keep it in the system. But when the tadpoles metamorphose into baby frogs, that's the weakest link. That's a really vulnerable time when the frogs develop chytrid in more of their skin. There's more surface area. Their immune system suppresses so they can grow new organs. And that is also reflected in our results, both in how many of those animals are infected, but more importantly, how severe and how intense those infections are. We can tell from the genetic results. But what we find most exciting and optimistic is that if you look at the difference between what's happening to the baby frogs and the adult frogs, although some of the adult frogs, so some of these babies make it. We don't know how and why, if they're just lucky or if they're genetically stronger to have a better immune system. But luckily, we still find some adults, but way less than what the books say used to be normal. But those adults we consider special because very much fewer of them are infected proportionally, but more importantly, it's the infection intensity. So the infection intensity in the adults is incredibly low. And through some of the radio tracking work that we've done, we've also seen animals, what we would consider self-curing. So we've had animals that tested positive, and then a week or two later tested negative. And even if it doesn't mean it's negative for chytrid, but it shows that over time, it's not developing disease.
Right.
So the strategy for HARC that we think could work is by taking out these weaker, younger animals before they die as they come out of the water. And then at HARC, care for their infection and disease progression. So it doesn't mean that we necessarily need to cure them or that we need to kill all the chytrid, because in nature, it'll never be gone.
Of course.
But our goal is to simply have the animals survive. And that means you can still have chytrid in the system, but there are things that you can test, such as reducing stress and improving nutrition, perhaps giving some heated basking areas that could suppress the infection. There's a wide range of things that we can try without having to revert to chemical therapeutics, which we can try if there's nothing else that's working. but basically boosting these animals, health and development to the point that they're older and stronger, and we decide that they should be going back into the wild. And I will also just mention, because I don't often talk about my partners, but I'm the most vocal person on social media compared to everyone I work with. But I mean, HARC is a huge team effort. There are multiple different institutions and organizations involved in this, and most notably, Omaha's Henry Doorly Zoo and Aquarium in the United States has been my longest time partner and funder for a lot of the work that we do. So we're always still looking for more partners, but Omaha has been huge.
That's great. Well, we got in a question through the chat that was related to what you were just talking about. So we had a student who was in Honduras with Opwell and saying that they learned that the eggs of the Croggaster rostralis frog develops within the adult. And they were wondering if this reproductive mechanism lowers the frog's susceptibility to chytrid during their most vulnerable stages.
That's a really good question, and I think we don't talk about that as much as we should. So Croggaster frogs are sometimes called rain frogs. These are direct developing frogs that lay their eggs on land and never need like a standing water body. So they're really special and unique in that the eggs just stay moist from the moss and the leaf litter and the rain, and then just tiny little baby frogs pop out. So these are the kind of things that I've explored in my work in Kasuko, because conventionally you would assume that if chytrid lives in the water, well then do frogs that not go to the water have less exposure to a disease event? And unfortunately, what I found, the answer is no, they're not safer. I've sampled a lot of those frogs and actually found quite a bit of chytrid in them, because I feel that chytrid is being carried away from the water by so many animals that they're basically dripping it through the forest. So essentially, nowhere in the forest is completely safe from exposure from chytrid. And that was part of the reason we also looked at the rainwater, because we were finding that even the frogs way up in the trees that live in the bromeliads, that, well, maybe they would be safe because that's just rainwater, and they were getting infected too. So no, I think these are really important lessons to keep in mind about not making assumptions from what we read in the books versus what we expect in the wild, and that this really matters for how we decide to prioritize what species need help. Because if you just read the books and said, okay, direct developing frogs never go into a water body, okay, well then they don't need help to protect themselves from an aquatic pathogen. And here we are in a rainforest with an aquatic pathogen infecting terrestrial animals.
Well, that makes sense, and obviously good kind of encouragement to people to question things that they read, right? I mean, everything that we know in textbooks is stuff that people like you were out in the forest looking at things and deciding and questioning how things worked out. So keep questioning, I guess. Yes. We're just about out of time, so I wanted to give you a chance if there's anything that we didn't bring up right now. Feel free to add any final comments, and then as well, if there's any kind of social media or projects that you're working on that people who are interested can go look at, feel free to shout them out so that people can keep following your work.
Okay. Two quick things. I know a lot of people were interested to know a bit more about the frog that was previously declared extinct that I rediscovered as part of my field work, called Croggaster milesi, the Miles rain frog. There are some articles online you can look up. I think there's one in Discover magazine a couple years ago called The Lazarus Frog. But just really briefly, yes. So this was actually one of those direct developing frogs that we just talked about. Yeah. And one of two species that had been formally declared extinct in Honduras at the time that I was working there. It had vanished in the mid-1980s. It was locally very abundant before then. But it was only known from two places, Kasuko and another national park. And then, 1 year the researchers go back and they're just gone. So it really added up as a good smoking gun suggestive case of chytrid fungus, because that's when around the time frame it was assumed to have swept through Central America in like the mid-1980s. But yeah, a couple years into my work in Kasuko, I did stumble across one. No, I had no idea what it was when I found it, but I knew it was important because it was nothing that I'd seen in the books. So reaching out to some experts and having some verification, it was easy to tell that it was different enough, and that it was this frog that had been declared extinct. Um, Whether or not that means that it's good, that it's, does that mean it's coming back from extinction, or is it still on the verge? We just don't know. I know some people have asked about, well, is that going to be part of a conservation program? At the moment, no. I know a lot of people think, oh, if you finally rediscover something that was thought to be extinct, well, like, of course you have to catch them and like save them. But, that's also a huge responsibility, and a lot of risk is involved with that too, especially when we don't know what it takes to keep them happy and healthy. Like, they've never been kept in captivity. So yes, it's enticing to want to, bring them into another species for hark, and maybe someday we will, but a lot of thought has to go into that. I'd rather know that they're still in Kasuko and hopefully hanging on before making some serious decisions about that.
And I guess it just quickly begs the question, did the ones that you have found in Kasuko, did you swab them for chytrid?
Yes, and they were negative for chytrid, which is great because they were in a stream where I know chytrid was present. But, a swab is one point in time. So with such a small number of swabs and a small number of animals, it's very optimistic. It's very encouraging. But we can't say too much about what that means as a whole.
Of course.
Yeah. And then the other thing, real quickly, I know some people were interested in science communication, which I haven't really talked about much.
Yes.
And just like if there's any main take-home messages for how to do it and how to do it well. And the one most important thing I'll say is be yourself, as corny as that sounds. I think especially for people in science, that showing your personality and not being afraid to talk about feelings and things you are excited about, and like expressing that part of you is what makes science communication effective. That's what helps break the stigma and the mold of what people think of as scientists versus what we really are, and it makes science more approachable. So yeah, be your own personality and have fun with it.
Good advice. Well, any last shout outs that you want to give to any social media or projects that you're working on?
Yes, follow me at My Frog Croaked, Twitter and Instagram. If you're interested in more videos on Hark, Hark has a YouTube page called Hark Frog Rescue. We have a few different video updates there. You can also check out our website, frogrescue.com. That we were very excited that that domain was available a few years ago because it's awesome. And lastly, citizen science. If you ever see a dead salamander, please Google and look up my iNaturalist citizen science page, called Saving Salamanders with Citizen Science. I know we talked a lot today and in my presentation about frog chytrid fungus, but there's also one now that's affecting salamanders, and it hasn't been found in North or South America, but we're terrified that it might show up. So if you ever see a dead salamander for unknown reasons, snap a photograph and upload it to my page. I don't need records of things where you know why it died. So things that were stepped on or roadkill, there's a lot of roadkill. But if it's something that just looks like it dropped dead or has sores or you see a bunch at once, those could be really important records.
Great. Well, perfect shout out. Thank you so much for your time this morning, John, and your great lecture on chytrid and amphibians and what it takes to, study something so in depth. So I really appreciate you taking the time to chat with all of our students, and have a great rest of your day.
Thanks. And yeah, thanks to everyone for joining.